Hello, my name is Marina Yurieva, and I'd like to present my work on visualizing the significant RNA binding motifs in alternatively spliced data. Alternative splicing is a molecular mechanism that modifies pre-mRNA constructs prior to translation. It results in production of proteins that vary in their sequence and activity and play important roles in development, cancer, and immune response. With the advances of technology, the estimated proportion of human genes that produce alternative mRNA isoforms increased to 95% of the genes. It can create hundreds and thousands of isoforms in multi-exonic genes. There are multiple splicing regulatory elements that are located in introns and exons. There are three core splicing signals present at each intron: 5' site, 3' site, and branch site. They participate in the splicing reaction. Splicing factors regulate splicing through splicing enhancers and silencers. They're usually located near the splice sites but can also be inside the introns. There are various splicing factors that can bind to both enhancers and silencers, and depending on that, activate or repress splicing. Various splicing factors bind and regulate splicing near the splice sites. Many of the RNA binding motifs of these proteins are known and their database exists. We have many bulk RNA-seq datasets and investigate alternative splicing in case control groups in them. And our goal is to develop the pipeline that also visualizes the RNA binding protein motifs in those datasets and estimates their significance. Previously, we developed a pipeline that identifies differential splicing events from our case control groups in bulk RNA-seq datasets using RMATs. The pipeline I'm presenting to you today takes the output of the RMATs, takes several groups of splicing events, and runs RBPmap, a tool that identifies RNA binding motifs within given coordinates of the genome. We apply Bayesian probability to identify significant events and then visualize them. This is our output of RMATs and the input for the current pipeline. The RMATs table is much longer, but we only using few columns from it. We need DPSI values that act as a fold change in splicing and show how many events get skipped or included in case control group. We create several groups of events for our analysis. Background events have less than 1% DPSI and are used for our Bayesian model. We split significant events into skipped and included, and because they will be plotted separately. After we filter the splicing events, we select the regions where the RNA binding motifs need to be identified. We excluded the splice site regions as they might bind too many proteins, but we select the regions around them. There are different types of splicing events, and for each type, the pipeline selects different set of coordinates. When we have the list of coordinates, we run RBPmap tool, which gives us the frequency of each motif at each position in the provided region. We are running background and significant samples in parallel, and different regions are getting processed at the same time as well, but we are running one protein motif at a time. RBPmap is located in a separate server that has a memory limitation, and we have the cutoff on the number of jobs submitted to the server so it doesn't crash. RBPmap provides a list of motifs the user can choose from, or the user can input their own motifs. After RBPmap step is done, we calculate Bayesian probability from the frequencies of the motifs per coordinate. The visualization shows frequencies for included and skipped events, as well as the background, and that is a gray line, a thin gray line here. And also we visualize the Bayesian probability for skipped and included events. You can also see that the frequency can be confusing and show the high number of events, while the number of background events is even higher. But with Bayesian probability, we can see the regions with the real significance. Here you can see skipped and included events in the iPS cells with deleted SRSF3 poison exon, and different types of splicing events that get affected by that deletion. And now we're in the process of creating a Docker container for this pipeline. I'd like to thank Anjukov Lab and Olga Anjukov for generating the data for the analysis and all their input, and our IT and computational sciences departments for their support. Thank you for your attention.